Hello my dear students, welcome back to Biology at Priyajit. In today's class, we are going to explore one of the most fascinating areas of microbiology, that is virology. Virology deals with the structure, replication, genetics, evolution and host interactions of viruses. But before we begin, let's consider a fundamental question. Are viruses rely living organisms? Viruses are acellular infectious agents that cannot reproduce independently outside a suitable host cell. They lack ribosome and complete metabolic machinery, but once inside a susceptible host cell, they can replicate their genome, produce viral proteins, assemble new virions, and sometimes destroy the host cell. In this class, we will cover viral structure and morphology, bacteriophages and the T4 lytic cycle, lysogeny, lysogenic conversion, induction, prions, viroids, and the biological significance of bacteriophages. So let's begin our journey into the fascinating world of virology. Let's begin. General characteristics of viruses. Viruses are generally acellular infectious agents. They contain their genomic material, which is either DNA or RNA, not the both. They do not possess a cellular organization. Their genome is surrounded by a protein coat called the capsule, and in some viruses there is an additional lipid membrane called an envelope. Viruses lack ribosome, independent protein synthesizing machinery, complete metabolic systems. They also do not reproduce by binary fission like bacteria. Instead, viruses replicate inside suitable living cells. This is why viruses are commonly described as obligate intracellular parasites. Another important characteristic is their small size. Most viruses are much smaller than bacteria and require specialized techniques such as electron microscopy for their visualization. Viruses also frequently show a high degree of host specificity and tissue specificity. In other words, a particular virus may infect only certain host organisms, particularly cell types within a host. Now there is important term even microbiology student must remember, that is virion. Virus particle outside the host is called as virion. Structural organization of virus. Viruses, the most and first important component is the viral genome. It can be either DNA or RNA, which is single stranded or double stranded, linear, circular or segmented. The diversity of viral genome is one of the most remarkable features of virology. For example, T4 bacteriophage contains double-stranded DNA. Bacteriophage lambda also contains double-stranded DNA. Adenovirus and herpes virus are also DNA viruses. On the other hand, influenza virus possesses segmented negative sense single-stranded RNA. Poliovirus possesses positive sense single-stranded RNA. Rabies virus contains negative sense single-stranded RNA. HIV contains an RNA genome and uses reverse transcription during its replication strategy. The viral genome ultimately carries the information necessary for the production of viral components and the formation of new virions. The capsid is the second major component is the capsid. Capsid is the protein cell surrounding the viral nucleic acid. It is made up of repeating protein subunits called capsomeres. The capsid performs several important functions. First, it protects viral nucleic acid from physical and chemical damage. Second, it helps the virus survive outside the host cell. Third, in some viruses, the capsid participates in attachment to host cell receptors. It can therefore contribute to host specificity. And finally, the capsid participates in the delivery of the viral genome into the host cell. The combination of viral nucleic acid and capsid is called nucleocapsid. Viral symmetry. Now let's move to the another important concept, viral symmetry. The arrangement of capsomeres give rise viruses characteristic structural patterns. Three major types of symmetry are commonly recognized. First, helical symmetry. Second, icosahedral symmetry. Third, complex symmetry. Let us understand the symmetry one by one. Helical symmetry. In simple viruses, capsid proteins are arranged in a helical pattern around the nucleic acid. The resulting structure may be rod-shaped, filamentous and flexible. A classical example is tobacco mosaic virus or TMV. In TMV, the RNA is associated with capsid proteins arranged helically. Icosahedral symmetry. The second major pattern is icosahedral symmetry. And icosahedron consists of 20 triangular faces, 12 vertices and 13 edges. Many animal viruses and bacterioviruses possess capsids showing approximately icosahedral symmetry. Examples include adenovirus, poliovirus and many bacteriophages. Why this arrangement is useful? Because it provides an efficient way of enclosing the viral genome using a limited number of protein types. Complex symmetry. Some viruses cannot be adequately described as either purely helical or purely icosahedral. These viruses possess complex symmetry. A classic example is the pox virus, which has a complicated multi-layered structure. Some bacteriophage also possess complex head and tail organizations. Viral envelope. Now let us discuss the viral envelope. Some viruses possess an additional outer membrane like structure called the viral envelope. The envelope is generally derived from a membrane of the host cell during the viral release, although viral proteins and lipids become incorporated into this membrane. Examples of enveloped viruses include influenza virus, HIV, herpes virus, and coronavirus. Viruses without an envelope are called non-enveloped or naked viruses. The viral envelope often contains viral glycoprotein that project outward as spikes. These spikes are extremely important. They may participate in attachment to host cell receptors, membrane fusion, entry into host cells, host specificity, and antigenic recognition. Therefore, when you study viral glycoproteins, always remember that they are not merely decorative structures. They are functionally critical components of viral infection. Matrix protein. In many enveloped viruses, a protein layer exists between the nucleocapsid and the envelope. This is known as matrix layer or matrix protein. Matrix proteins stabilize the virion, connect the nucleocapsid with the envelope, participate in assembly, and influence viral budding. Therefore, the matrix layer acts as an important structural and functional bridge between the internal viral components and the envelope. Viral enzymes. Some viruses carry enzymes within the virion itself. Why? Because these enzymes are required immediately after the virus enters the host cell. Examples include reverse transcriptase in retroviruses, RNA-dependent RNA polymerase in many RNA viruses, certain proteases, other enzymes involved in genome replication or processing. This tells us something very important about viruses. Although viruses depend heavily on host cell machinery, they are not completely passive. Some viruses carry specialized enzymes that the host cell cannot provide in the required form at the required time. General viral morphology. The virus may be spherical, rod-shaped, filamentous, bullet-shaped, brick-shaped, complex head and tail structures. TMB is rod-shaped, influenza virus is spherical or pleomorphic, rabies virus is bullet-shaped, pox virus is complex or brick-shaped, and T4 bacteriophage is complex head or tail structure. This is why viral morphology becomes much easier to remember when you associate each shape with a representative example. This is the different types of viral morphology. Bacteriophages. Bacteriophages are simple phages are viruses that infect bacteria. The word bacteriophage literally means a bacteria eater. Bacteriophages are enormously important in microbiology. They have contributed to bacterial genetics, molecular biology, microbial ecology, and bacterial evolution, phage therapy, and biotechnology. Two major reproductive strategies of bacteriophages are the Lytic cycle and the Lysogenic cycle. The best way to understand these pathways is through two classic examples. For the Lytic cycle, we will discuss T4 bacteriophage, and for Lysogeny, we will discuss lambda phage. Structure of T4 bacteriophage, which includes head, neck, tail sheath, base plate, tail fibers, and tail pins. Lytic cycle of or T4 bacteriophage. The steps are attachment or adsorption, penetration, and injection of the viral genome. Early phase of T4 infection, then host DNA degradation, T4 DNA replication, late gene expression, assembly of T4 phage, maturation, and lysis of the E. coli cell. Thus, the Lytic cycle is complete. This is the image showing the Lytic cycle steps. Lysogeny. The classic example is bacteriophage lambda infecting E. coli. Such a phage is called temperate phage. The steps are prophage and lysogen. The lysogenic pathway of lambda phage. The first is the lytic pathway. The second is lysogenic pathway. In the lytic pathway, viral replication, then assembly, then lysis. But in lysogenic pathway, integration, prophage formation, replication within bacterial chromosome. So this is the image showing the lysogenic cycle of the bacterial cell within bacterial cell. Establishment of lysogeny following infection, lambda DNA enters E. coli and circularizes under conditions favoring lysogeny. The phage genome integrates into the bacterial chromosome. This occurs through site-specific recombination. The integrated prophage is then replicated along with the bacterial chromosome. Whenever the bacteria divides, therefore, one lysogenic bacterium, daughter cells carrying the prophage. This allows the phage genome to persist for many generations without immediately killing the host. Maintenance of lysogeny. A central regulator of lambda lysogeny is the CI repressor. The CI protein represses genes required for lytic development. As long as repression is maintained, lytic genes remain largely inactive. The prophage remains integrated. The bacterium survives and reproduces. The prophage is inherited by daughter cells. This represents a highly controlled balance between phage and the host. Lysogenic conversion. So therefore, the bacteria can contribute directly to bacterial virulence. Significance of lysogenic conversion: first, it can increase bacterial virulence; second, it contributes to bacterial evolution; third, it can influence pathogenicity; fourth, it increases genetic diversity; and fifth, it can influence the epidemiology of bacterial diseases. This is the image showing the steps of lysogenic conversion. Induction of prophage. A lysogenic state is not necessarily permanent. Certain environmental or physiological stresses can cause a prophage to leave the lysogenic state and enter the lytic pathway. This is called induction. Common inducing agents are ultraviolet radiation, DNA damaging chemicals, certain types of cellular stress, conditions that activate the bacterial DNA damage response. So classical mechanism involves SOS response in E. coli. Molecular basis of induction in lambda phage. Under normal condition, the lambda phage is maintained by CID pressure. When DNA damage occurs, the bacterial SOS response is activated. A key regulator protein is REC-A. Activated REC-A promotes the auto cleavage and inactivation of CID pressure. As CID pressure is lost, lytic phage genes become active. So the prophage then proceeds through excision, viral DNA replication, late gene expression, assembly, and lysis. The biological significance of induction. The image showing the steps. The difference between lytic and lysogenic cycle. Prions. Viruses, not only the infectious agent of interest in microbiology, prions represent an entirely different category of infectious agent. The term prion is derived from proteinaceous infectious particle. A prion is an infectious agent composed essentially of misfolded protein without a conventional DNA or RNA genome. This concept was particularly associated with the work of Stanley B. Prusiner, who proposed the prion hypothesis. Structure and nature of prions. The normal cellular prion protein is commonly designated as PRPC. It is a host encoded protein. The abnormal infectious form is designated as PRPSC. The abnormal protein has a greater proportion of beta sheet structure compared with the normal form. The abnormal conformation form can promote conversion of normal PRP into abnormal conformation. The simplified model is PRPC into PRPSC. The abnormal protein accumulates and associated with progressive neurodegenerative disease. Diseases associated with prions are in humans: Creutzfeldt-Jakob disease, variant CJD, Kuru, fatal familial insomnia, Gertzmann-Strauss-Richter syndrome. In animals, scrapie, bovine spongiform encephalopathy, chronic wasting disease in cervids. These are the differences of viruses and prions. Viroids. Viroids are another unusual category of infectious agent. They are much simpler than viruses. They are essentially a small circular single-stranded RNA molecule that lacks a protein coat or capsid. The viroid is naked infectious RNA. Viroids are primarily known as pathogens of the plants. Structure of viroids. They are small, circular, single-stranded, highly base paired, non-coding in conventional strains. They are not enclosed within a protein capsid. Examples include potato spindle tuber viroid, Citrus exorcotis viroid. Replication of viroids. It is host cell enzymes it utilizes. It follows the rolling circle model. The virusal replication is interesting because the viroid RNA does not encode its own complete replication machinery. This is the differences of virus and viroid. Why are viruses considered unique? Because they can express genes, replicate their genome, synthesize proteins, assemble new particles, alter host metabolism, sometimes kill the host cell. So they are often described as obligate intracellular parasite. So this is the difference between viroids, viruses, and prions. So which are the distinct infectious agents? Virus contains nucleic acid, viroids are naked RNA, and prions are misfolded protein. T4 phage follows the lytic cycle, while the lambda phage can establish lysogeny. Together they demonstrate how viruses influence host physiology, evolution, and pathogenicity. That brings us to the end of our discussion on virology. Keep exploring, keep questioning, and keep learning, because every microbe has a story to tell. See you in the next class. Bye. Thank you.